بسم الله الرحمن الرحيم وبه نستعين اللهم علمنا عنك يا رب فهمنا عنك يا رب. Okay, in the first day of my journey to learn about bioinformatics, okay, in the last of the day he gave me a task. Okay, this task it will take nearly 15 minutes. It contained that I need to read a short article or watch a video explaining the central dogma of molecular biology. And the second task was think about how analyzing DNA and the RNA sequence can be useful for medicine or research. So I convert this thinking into a question to ChatGPT to expand my knowledge. And this what he gave me. Okay, he said the following: analyzing the DNA and RNA will help you in understand the genetic disorder, the infectious disease. Okay, analyzing the DNA sequence unveil mutation. So when I analyze the DNA, I can detect the mutation that predispose individual to disease. Okay, and he gave me example: mutation of the gene of PRCA gene can indicate elevated risk of breast cancer. So once I know this, I can give the patient advices about she need to come more frequent to be examined. There is another option of surgeries. Okay, so we start earlier to deal with the with the disease before it become already present with us, which is meant by called the preventive measures. Okay, what else related to infectious disease? I will take advantage of DNA or RNA sequencing. We can sequence the RNA of the virus like SARS, COVID-2, enabling rapid identification and tracking for these pathogen. What is the benefit? I can facilitate effective disease control measures. Okay, what else? Personalized medicine. Okay, what is meant by personalized medicine? It is tailoring treatment for each individual. In our protocol in medicine, we make what is called one size fit all. Okay, we have this protocol for each patient with this stage of disease. But in personalized medicine, no, we tailor this protocol for this person according to its pharmacogenomics. Okay, according to specific genetic marker inside it. Okay, what is the benefit of this tailoring of disease? We can increase the efficiency of the drug response and decrease the complication. Pharmacogenomics, it is we do DNA analysis for this patient, allow us for apply this personalized drug dose, considering how individual gene can influence the drug metabolism. Some is responsive to the drugs so they didn't need a large doses. Why they take it and have complication? Okay, this ensure optimal treatment outcome and minimize the adverse effect. What else? Cancer therapy. Identifying mutation in tumor. It will provide us insight for targeted therapy, drugs that will work on this specific problem. This lead to increase the efficiency of treatment and improve the patient outcomes. What else? We can understand the gene function. DNA analysis will give us a proper understand for the gene function. Okay, on what level? Number one, gene expression. What is meant by gene expression? Gene, it is a sequence of nucleotides. These sequence of nucleotides on the DNA carry information. This information need to express itself. Express itself in what mean? Producing a functional product. This functional product may be protein or RNA. Okay, this is what mean by a gene expression. Okay, so when I understand how many of this functional product is produced from a gene, okay, through RNA sequencing, it will reveal which gene are active. What is meant by this? Each cell have the whole set of genome, but which gene is expressed in this specific cell, okay, and in which context? What I mean, a liver cell. Liver cell will express the gene, okay, that produce albumin, okay, while the skin cell will not produce this gene, will not express the gene. So when analyze or I sequence the RNA in each cell, I will found the RNA, messenger RNA of albumin in the liver cell, but I will not found it in the skin cell. So I can understand what a set of genes are active in each cell, and these genes, I understand that they have a function in this specific cell. At the first level, I know or I learn about the expression under normal condition. Then I search about the expression under pathogenic condition, so I can understand the mechanism of the underlying disease. Okay, so he said this RNA sequencing reveal which gene are active in a specific condition and a specific tissue, which genes are active in liver, which are in the skin, and the specific condition including in normal condition and in diseased condition. This provide valuable insight in the molecular mechanism underlying the disease. And once I understand the molecular mechanism, I can I can design drugs to these targets. Okay, what else? Cellular process. Studying the RNA sequence help unreveal how gene regulate the cellular process, the apoptosis, the cell cycle, okay, the metabolic pathways, and contribute to the development of disease. And which are these processes, okay, are affected in a specific disease mechanism. So you understand the process, okay, in the cellular process, which RNA are affecting these process, and the disease that which process are affected. Research application. Understanding the gene function will facilitate research in area of drug discovery, disease modeling, that I can make a more similar animal model for the disease. Okay, I know the gene that is disrupted in this pathogenic pathway, and I design animal model and through disrupting the same gene. And at the last, personalized medicine. I can target drugs to this specific problem. Okay. He will focus on the process of drug development as a benefit from RNA and DNA sequencing. Okay, so he will make the story of the drug development from bench to bedside, from the first identification to already to the clinical trial, and how RNA sequencing help us in this discovery. So is it the following: sequencing help discover new drugs, targets by pinpoint gene or protein crucial for disease process. Drug design and development, understanding the molecular mechanism which we obtained through sequencing. Okay, on the normal set and on the disease set, enable us to design and development a target therapy, which potentially lead to what? A more effective, a more safe treatment. Once we design this drug, we start in our clinical trials. Once a drug candidate is identified, clinical trials are conducted to evaluate its safety and efficiency in human. This paving the way for more faster approval and availability of treatment for disease. Okay. Another story that or another aspect that it help us RNA sequencing and DNA sequencing in researchers in it is evolutionary insight. Okay, tracing our ancestor journey. We can use this sequencing of DNA and RNA in comparative genomics, in doing a phylogenetic tree. Okay, in doing human ancestry and population genetic. Okay, what is meant by phylogenetic tree? It compares the DNA sequence across different species, reveal evolutionary relationship, and construct a phylogenetic tree that illustrate the history of life on Earth. Okay, human ancestry. DNA analysis help to trace the human ancestry, identifying genetic markers that link individual to specific geographic regions and ancestor population. Population genetic, studying the DNA sequence across population, it will provide insight into genetic diversity, migration pattern, impact of natural disasters in evolution. How analyzing the RNA and DNA sequencing can making a revolution in health care. Okay, number one, through personalized diagnosis, targeted therapy, and preventive medicine. Okay, what is personalized diagnosis? DNA and RNA analysis enable personalized diagnosis, leading to earlier detection. Okay, we analyzed the DNA and the RNA of this patient. We found a specific marker that indicate a increased risk of specific disease. Okay, leading to a more effective treatment strategy for this disease. Targeted therapies. Identifying specific genetic markers allow for targeted therapy, increasement in treatment efficiency and reducing the side effects. Preventive medicine. Predicting genetic testing empower the individual to take a proactive step in mitigate disease risk and improve this overall health. At last, what is the future of medicine? The future is that we will develop a precision medicine. We will use this RNA and DNA sequences in disease prevention, in forming a new therapy. In precision medicine, the DNA and RNA analysis will play a key role. Development of a precision medicine, tailor treatment to individual patient. In disease prevention, advancing in genetic testing, understand the gene function, will empower preventive medicine, minimize the burden of the disease. New therapies, continued research and technological advancement will unlock novel therapeutic approach, potentially making a revolution in treatment of currently incurable disease. Thank you. سبحانك اللهم وبحمدك أشهد أن لا إله إلا أنت أستغفرك وأتوب إليك.